Thanks very much. Good morning, everyone. Today I'm going to be talking about some work I've done on looking at determining the origin of Phragmites australis in South Africa. Phragmites australis, otherwise known as the common reed in South Africa, is a perennial reed-like grass. It's a clonal grass and it reaches heights of about 4 meters. Phragmites is considered a cosmopolitan species. It's actually considered one of the most widely distributed angiosperms in the world. And this is largely due to its high plasticity. The reed is highly adaptable. It can be found in freshwater ecosystems all the way up to brackish, tropical regions all the way up to temperate regions. So it's a very highly adaptable plant. In South Africa, Phragmites is considered an indigenous species, and this is largely based on evidence of a long history of utilisation. It's traditionally been used for thatching, for building, so it's considered a very important plant in South Africa. And it's very widespread. This is just a very, I'm sure, quite out-of-date map on the distribution of Phragmites. It's just to show you that it's got a very wide distribution in the country. It's present in all provinces. However, quite interestingly, in recent years there's been quite a dramatic increase in the abundance of Phragmites in South Africa. We've seen quite a dramatic increase into even Phragmites spreading into previously unoccupied habitats. And there's been quite a bit more research now done as to why this is happening. And just to give you an example, in the wilderness lakes, Phragmites australis has always been a component of this ecosystem. However, from about 1975, there's just been quite a dramatic increase in the reed stand, and it's becoming quite a lot more abundant. And this has become quite debatable. Some people are saying it can become quite problematic due to this sheer size of these reed stands. And just to show you an image, this is taken of a Phragmites stand in the Western Cape along the wine region. And you can see here, this is quite a dense stand of Phragmites. And then here by Gullis Bay, another example of a Phragmites stand. So further research is necessary to just look further into this, and this highlights a similar case in the US. They actually have a very similar thing happening over there, where Phragmites australis is considered native to the US, also a long history of utilisation. And what was happening in the last 100 years was that there was this dramatic increase in reed stands. And reed stands were becoming actually quite invasive and sort of taking over a lot of wetland areas. And this spurred on a lot of research into why this was taking place. Why was this native plant all of a sudden taking over? And a lot of studies were done looking into why a plant was suddenly becoming invasive, and this included anthropogenic reasons, rivers changing, could that be the reason? And one of the components was looking at the genetic factors as to why a plant would suddenly become invasive. And a study by Saltonstall in 2002 found that by looking at the genetic haplotypes, so the genetic strains that were present in Phragmites populations in the US, it was found that the stands that were actually becoming invasive and moving into new areas actually had a different haplotype to stands that were not invasive. And what ultimately was found was that the stands that were invasive actually had an introduced strain that was actually originated in Europe. So what was quite interesting about this was that the introduced haplotype was acting much like an alien invasive. It was moving in and it was displacing native vegetation. And just to show you a very clear depiction as to what was happening, on the left here, this is the introduced haplotype of Phragmites. And then this is the native Phragmites, which is still considered native to North America. And you can see here, it's actually a very clear morphological difference as well. The invasive haplotype is a lot taller, it has larger flower plumes, and this has actually made it a much better competitor if you will, to the native Phragmites. And that's what's causing it to sort of largely take over and displace native Phragmites stands. So what I sought to do in this study was to determine the haplotypes that we have in South Africa and then compare them to worldwide haplotypes. And this would ultimately help us to understand the origin of our Phragmites stands and whether or not we have a similar case that's happening in the US. And this was done by collecting leaf samples from across South Africa. This is a map here of sites taken, of my sample sites. I tried to get as much of the distribution of Phragmites as possible. DNA was then extracted, and I followed Saltonstall's protocol and amplified two non-coding regions in the chloroplast DNA. And this was the TRIN region and the RBC region. These regions were then sequenced and compared to worldwide haplotypes on GenBank. This study is actually still in progress. It's not yet finalized, but what I do have is my sites from the TRIN region. And what I can report on so far is it's actually very interesting. There's actually been no genetic variation from my Phragmites sites, both within populations and across sites in South Africa. And that all of the TRIN sequences matched up with what Saltonstall has labeled haplotype 3. And this haplotype 3, she's labeled as the unique African haplotype. And this is based on the fact that it's very widespread in southern Africa. Haplotype 3 is found in Botswana, Namibia, and South Africa. But as I said, this is not finalized, these results, as I still need verification from the RBC region. Haplotype 3 is labeled as an African haplotype. However, quite recently, a study by Lambertini in Europe in 2002 found haplotype 3 populations of Phragmites in Spain. So now this raises some serious questions on the validity of calling haplotype 3 a unique African haplotype. We need to ask the question, is Phragmites a native plant, and was it simply introduced to Spain quite recently, or was Phragmites in fact an introduction from Spain? And there's a number of ways that we can look further into this, and I do plan on doing so. One of the ways is looking at the genetic diversity of our Phragmites populations. This has been done in Europe. They've looked at microsatellite diversity as an indication of genetic diversity. And I plan on doing microsatellite analysis and comparing the results with the European genetic diversity. And what this will show is that if we have a low genetic diversity in our Phragmites populations, this will show us that our Phragmites is most likely not a native plant and was in fact introduced from Spain. If we find that there will be a higher genetic diversity, then we will have a lot of evidence to show that Phragmites can still be called a unique African haplotype 3 can be called that. Another way of further investigating this, which I'm also planning on looking at, and it's also in progress at the moment, is looking at the herbivores associated with Phragmites. As we all know, the diversity of herbivores on a plant gives a very good indication on the origin of a plant. And that's just from looking at how the herbivores evolved to feed on the plant. What I have found so far is that I've only found 3 herbivores that are abundant, widespread, and damaging on Phragmites. And this is surprisingly low for a plant that we consider a native to South Africa. And what's more interesting is that this actually is very similar to the results they found in the US. A study by Tweeksbury in 2002 found that the herbivores associated with Phragmites, there was only about 5 native herbivores feeding on Phragmites in the US. And if we compare this to similar studies in Europe, there's been 140 herbivores found feeding on Phragmites. So already this is starting to raise some serious questions as to why this is the case. Why are we having a lot more herbivores feeding on Phragmites in Europe and so little feeding down here? But this is also still in progress, so it'll be interesting to find out. So just to conclude, there's still quite a bit of work to be done here, but what I will plan on doing is finish sequencing the RBC region, looking at microsatellite genetic diversity, finalizing looking at the herbivores associated with Phragmites. And all of these components together will give us a very good indication on the origin of Phragmites in South Africa, and we can then look further into what this means for our Phragmites. Thank you very much.
Any questions?
Not a question, just a suggestion. I think don't use the RBCL region. It's not going to help you. At species level, it's not going to work.
Well,
you won't get the resolution you're looking for.
The reason that the RBC region is so important is what Saltonstall did is that she has used both, and basically she combines the RBC region and the TRIN region to define haplotypes. So basically, it's sort of, that's how she has defined all her worldwide haplotypes. And so basically the TRIN region, it's called haplotype 3, but then when it's combined with the RBC region, it becomes haplotype K. I don't know if that makes sense. So it sort of needs the RBC region to verify its haplotype.
Yeah, well,
that makes sense.
You know, it's used at generic level.
Yeah.
So at population and species level, I'm not so convinced.
Yeah.
But that's also why Kim is doing microsatellites.
Yeah. Yeah. Just to go further into it.
Okay, let's give that to Kim.
Yeah, Kim, thanks for the very interesting talk. And well, just a comment first of all on the question that was raised there. I think they could still be done, and it's quite interesting because just to build up the haplotypes, that region is also quite informative. And then just my question was just on microsatellites. So I don't know, are you planning on developing new markers, or you just want to take the markers on gene bank and use the primers that they have developed already in
For me, I am going to sort of piggyback on what they've done in Europe, because the idea is to really just be able to compare what they're finding, so that it gives a very clear indication on the populations in Europe versus our population. So I will be using their markers, which have already been developed. Yeah.
Just two thoughts. I mean, one of which is primarily around the dispersal. If it is a European kind that got here somehow, how? And got so widespread in the time. You know, we have a lot of migratory birds that move between the northern hemisphere and the southern hemisphere. Is it feasible that they could have transported seeds with them? That's because they would go to most wetlands, and that might be then a route. It's just interesting. And then a second sort of related question will be over what kind of timescales could genetic diversity develop? You know, we've got a huge variety of regions across the country, so one would expect some genetic diversity within the species, also because of pollination ranges and all that kind of stuff.
Yeah.
Population differentiation. So just a thought.
Yeah, there has actually been a huge amount of work done on Phragmites and its dispersal, and they've actually shown, the Phragmites, because it's also, seeds can be dispersed so far with migratory birds, and it's also the fact that it's been so prized and used, that they've shown that it really is almost at a global scale now, that its dispersal. So yeah, I mean, there's so many ways in which it could have been introduced, and there's a lot of evidence behind that. And sorry, your second question?
The second question was what sort of time scale? So if you find that there's more homogeneous genetically here, over what kind of time scales do things actually differentiate?
Yeah.
And herbivores adapt, or whatever, those sort of time scales. Are we talking about millennia? Are we talking about millions of years? Just curious.
Yeah.
Make a guess.
I'm
Thanks. Thank you very much.